G6PD में क्या करना है आपको कि ये जनरली मेट हीमोग्लोबिन, ये भी कलरेशन का ही टेक्निक है, ठीक है। और ये कन्वर्ट करता है, जैसे सोडियम 3 सॉल्यूशन यहाँ पे यूज हो रहा है। वन इज सोडियम नाइट्राइट, अनदर इज द मिथिलीन ब्लू एंड थर्ड वन इज द ग्लूकोज। इन सम पेपर्स द सोडियम नाइट्राइट एंड ग्लूकोज आर कंबाइंड टुगेदर। ये सॉल्यूशन में क्या होता है, कंबाइंड होता है, तो वहाँ पे बोलेगा सोडियम नाइट्रेट ग्लूकोज सॉल्यूशन, ऐसे बोलेगा, ठीक है। तो इसमें इस टेस्ट के अंतर्गत करना क्या है कि 3 टेस्ट ट्यूब आपको लेना है। थर्ड यू सेलेक्ट द 3 टेस्ट ट्यूब्स एंड मार्क एज द टेस्ट ट्यूब, टेस्ट T पॉजिटिव कंट्रोल, P पॉजिटिव कंट्रोल, P and negative control, N. And negative control, ठीक है। And commonly the sodium nitrite is responsible for the conversion of hemoglobin into methemoglobin, ठीक है। And the color of the methemoglobin is brown, clear। And the methemoglobin when passes through the PPP system, it converts into the again hemoglobin in the presence of methylene blue. If the NADH is available in the form of NADH2, okay? And that NADH2 is produced by the enzyme glucose 6-phosphodehydrogenase, clear? So if it is not converted, if the methemoglobin is not converted into the hemoglobin, that means the enzyme glucose 6-phosphodehydrogenase is deficient in the RBC. Of course, RBC is present in the, glucose 6-phosphodehydrogenase is present in the RBC. So the lysis of the RBC is also necessary, which we use for the lysis, complete lysis of the RBC, we use the water bath technique, मतलब heat technique, heat and shock technique. And for the purpose, at first you we take the 0.1 ml or 100 ml of sodium nitrite, in test and in positive control tube, in test and in positive control tube. Now we take the 100 ml or 0.1 ml of glucose solution, again in test and in positive control tube. Then we use methylene blue. Then we use the methylene blue only in test. And then we Blood, where is blood? Take 1.5 to 2 ml of blood. Here, this one. In sorry. In test, where is tissue paper? Tissue. In positive control and in negative control. Okay? Now you mix it well. Now you mix it. Now you mix it. And here it is only blood. Then you put all these 3 tubes. Where is tissue paper? All these 3 tubes you put in hot water and set it up to the 35 degree centigrade. 30 at the 35 centigrade, put it in the hot water bath for the 90 minutes. रोक दो. Now remove the tubes from the hot water bath. Mix well. And clean. Dry the tubes with the tissue paper. Give me the pipette. Again take the 3 test tubes and Take 10 ml distilled water in each. Use the measuring flask and take 10 ml of distilled water. Okay. Okay. Keep all these three in holders and then take 1 ml of taste, 1 ml of positive control, in another tube, and 1 ml of negative control in other tube. Now mix. Mix well. See here. And then compare the color of taste with positive control and with negative control. If the color of taste represents similarity with the positive control, it represents that the glucose 6 phosphodehydrogenase is deficient, and if it resembles with the negative control, it represents that the concentration and value of the glucose 6 phosphodehydrogenase is normal. But in this condition, you see that the color is in between the positive control and negative control. It represents that the value of the glucose 6 phosphodehydrogenase is not normal but must in deficient condition. See here the color. You see here the color. The positive control represents the color of methemoglobin, whereas the negative control represents the color of the normal hemoglobin. But the tube of the test represents color in between the positive control and negative control. It may represent here. Here we observe that the sample may represent the deficient concentration of the glucose 6 phosphodehydrogenase enzyme, and at present it may be caused due to the delaying of the test, because we doing the work or the laboratory work after the 12 or 14 years of collection. It is necessary to carry test as early as possible after the collection of blood sample. You clearly observe here that the color of the test is methemoglobin, because we use here sodium nitrite solution and glucose only. But in the condition of test, we use the sodium nitrite, glucose, and methylene blue. And here the methemoglobin converted to the sodium nitrite is again comes in the hemoglobin, normal hemoglobin, due to the presence of NADH2 and methylene blue solution. So the color represents similarity with the negative control if the concentration of the glucose 6 phosphodehydrogenase is normal in concentration. But when it is deficient, then it resembles the color with positive control. See here, you clearly see here that here the color is dark brown, and in the present condition we observe that the enzyme glucose 6 phosphodehydrogenase is deficient, either due to the absence of the NADH2, or it may, in the present condition, just I am saying, that it may be caused due to the delay test. But the test is confirmed that The G6PD is deficient when it resembles color with the positive control, whereas in the normal condition it represents the similarity with negative control. Now it is we observe that the glucose 6 phosphodehydrogenase causes hemolysis, or in newborns it may be one of the cause for the development of the neonatal jaundice. Here you observe that the three samples we use, the nitric sodium nitrite, that is 1.25, 12.5% sodium nitrite, and the methylene blue solution represents the 0.0004 m strength, whereas the 5% glucose is used for the G6PD test here. Now, the G6PD test is significant for the population study, as it represents that the trait is associated with the protection from the malaria infection, as well as, on the other hand, it causes the hemolysis during the oxidative stress, and particularly when any person ingests the fava bean. Here we clearly see that the color in the taste, in the positive control and in the negative control is directly associated with the concentration of enzyme glucose 6 phosphodehydrogenase. Here we observe the deep brown color in the taste, and it represents that the glucose 6 phosphodehydrogenase is not sufficient as to convert the methemoglobin into normal hemoglobin. So in the test, the color resembles with the methemoglobin in the positive control tube. In the negative control tube, we took only the blood hemoglobin, so it represents the normal red color, whereas in the deficient condition it represents the deep brown color. And it is clear that the taste is depended on the coloration and decoloration principle of the methemoglobin. Now, as I think you all will